This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 41 for July 17th, 2009. Hi, everybody. It's Vincent Racaniello, and it's time for TWiV. And today we have a stellar cast assembled for you to talk all about viruses, starting with, here in my office, Professor Dick Despommier. Welcome, Dick.
Thank you, Vince.
You have a good week?
Had a great week.
Any fishing?
I did, actually. Yeah, it was fishing last, dare I say it, during the week sometime.
No, you don't work. You take a day off and you fish.
That's what I did.
Excellent. And we also have today Alan Dove from the wilds of western Massachusetts.
Yes, indeed. Good to be here.
How's the new house, Alan?
It's nice. It's, fun going through the list of 1,000,001 things that need to be fixed or adjusted. But,
excellent. And then we have, normally from Gainesville, but not today, Rich Condit. Welcome back, Rich.
Great to be here. Thanks very much.
Rich is in Oregon somewhere, right?
Yes, indeed. Just a little south of Bend in a town called Sun River.
Wow.
It's terrific here. It's beautiful. High desert.
Right.
Wonderful.
And not too far away from the Metolius either.
Right.
And by the magic of Skype, of course, we're all joined in clear stereo sound to talk about viruses. But Rich, you were just at a wonderful virology meeting, right?
I was indeed. The 2009, it's the 28th annual meeting of the American Society for Virology, held this year in Vancouver, British Columbia.
That in itself is beautiful.
Yep.
It's a wonderful place. It's just gorgeous. And we have we met an old friend that I haven't seen for 30 years. He was a postdoc advisor, Joe Cates.
Yeah, sure.
Who did a lot of the early work on pox viruses. I hadn't seen him for a long time, and he now lives in Vancouver. So he showed us around, and it was terrific.
Now, correct me if I'm wrong. Did Joe Cates discover DNA-dependent RNA polymerase?
Yes, well, at least virion encapsulated DNA-dependent RNA polymerase and poly-A. He doesn't quite get the credit for that, but he discovered that there was poly-A attached to vaccinia messages made in the virion core. He was in competition with some other people doing it in cells. So, but he did. At least independently, he discovered that.
Cool. So the meeting was great, right?
Meeting was terrific. I really enjoy those meetings. They're a great place to learn a great variety of virology and see a lot of people, put faces with names, and also concentrate on your own area of interest. Pox viruses were very active there this year.
Well, next year, TWiV will be at ASV. The council has approved us doing a live version in front of an audience there.
And I think that's terrific. That'll be fun.
And I'm looking forward to introducing the podcast format to all those virologists, especially the young ones, to get them interested. And we'll grab a bunch of people at the meeting and put them on stage, and wewe'll record it. And that'll be
in another good fishing location, right?
In Bozeman,
Absolutely.
Montana.
Absolutely.
I'll just sashay off the stream and show up.
You could show up in your waders, right?
That's what I meant.
No problem.
You really should have hip waders for the show.
Anyway.
Okay. We're all in good form today, or I should say Alan is in good form. You're in a good mood, Alan?
Yes, I had a good week.
All right. Well, we have a ton of stories today. I tried to find non-influenza stories, and there are a bunch of them. There are also a few flu stories. Since you joined us, Rich, the swine flu thing exploded.
Right.
And we have had just about every episode a good amount of swine flu information. What's the story in Florida? Has it been a big deal?
Nah, not a big deal. Every now and then you see a couple of cases reported in the newspaper. I don't think if there's been any deaths from it, I'm not aware of it. Mostly it's, so-and-so we now learn had swine flu and they're doing just fine.
Right.
You've
had more deaths from monorail collisions than you have from...
That's right. Be careful at Disneyland.
So Florida, according to CDC, has had 2,000 confirmed cases and 12 deaths.
Oh, okay.
Which is not the biggest in the country, but obviously it's not making the news since you live there and you haven't heard about it.
No, it's not been a big deal in the news.
Who is the leader then, Vince?
Which state is the leader?
Yeah.
All right. The state leading in number of confirmed cases would be Wisconsin at 6,000.
Wow.
Wow.
That is an unusual thing. I would have guessed California.
California has 3,000. Now, remember, these are just lab confirmed.
Yeah.
There are probably
And
plenty more, right?
Is that partly because Wisconsin has a particularly virologically oriented public health department?
Yes, it might be. Absolutely. I think every state doesn't have equal number of labs that are doing the diagnosis, and so it's really a bias. I've seen estimates that there are probably 1 million or more infections in the U.S. Most of them are not diagnosed. We won't really know until next year, probably.
They did make a big point out of the cadets that caught it, though.
Yeah, but it seems to be now that whenever you have big numbers of people gathering, it's a great venue for spreading infection.
Yeah.
So summer camps now are an issue. Lots
Sure.
of kids together, cruises,
Cruises,
things that
that's right.
happen in baseball games, sport events.
Virology meetings.
Yeah, it could be virology meetings as well.
Rich, that would be a twist of irony that would be palpable, in fact.
That'd make a great headline.
Wouldn't it?
Virology meeting struck by virus, yeah.
So, what are the data in humans so far? I realize these are slippery numbers, but what are the data in humans so far regarding the virulence of this flu relative to previous strains?
This is a very contentious point because the numbers are slippery. You know, we only have these confirmed cases so far. And typically, you do a lot of statistics to figure out mortality every year for flu, at least, because it's a very hard disease to pinpoint death to.
Mm-hmm.
But if you so far look at the confirmed cases and the deaths, it's quite low. It's 0.4%. Well, actually, that's high. It's higher than seasonal flu, slightly higher, but not as high as pandemic flu.
Right.
So a lot of people are saying it's relatively mild. In fact, we have an article today that we're going to talk about that.
But Vince, the WHO called this pandemic flu.
Well, that doesn't mean it's virulent, right?
No, of course not.
It only means that it's spread to so many countries.
Which is why I brought it up.
Yeah, I mean, let's talk about this in a couple of papers. We have a Nature paper that addresses this issue. My feeling is, from what I've seen, it's relatively mild, and the number of cases is only going to increase because we're only doing lab confirmed cases. So at some point we'll do statistics and figure out the total number of infections, and I suspect that at least now it's pretty mild. But not everyone will agree on that. There's lots of a discussion. But let's get back to that. Let's first go to salmon.
Right.
Yes.
Here's an interesting article. Walmart stops buying Chilean salmon because of a virus outbreak.
Which one?
It is called Infectious Salmon Anemia virus.
Salmon. Yeah, that's what I thought you'd say, actually.
ISA.
That's everywhere, by the way.
So Chile is a big producer of salmon. They farm it. Anybody know about salmon farms?
Yes.
I know a lot about it.
Tell us a little bit about it, because I'm not an expert on salmon farms.
Well, who wants to go first?
Oh, you go ahead. You go first, Dick.
All right. The salmon that's being farmed is Salmo solar, which is the Atlantic salmon, which can actually spawn more than once in its lifespan. The Pacific salmon spawn only once and then die. So they live a longer time at sea before they come back. So the Atlantic salmon appears to be a more culturable, domesticational salmon species. So that they have picked that, and because that's the one that most people eat on the East Coast and throughout Europe as well.
So what is a salmon farm?
The salmon farm is an enclosure in the ocean, very large, with a big net underneath it. And the net is of various sizes, depending on the size of the salmon yearlings that they've got. It takes about 1.5 years to grow up a full commercial length salmon of about 6 to 8 pounds, which is what's sold at the markets. The flesh is basically fatty compared to a normal wild salmon because they don't exercise much, so that they're not looked at as the best source of all these omega-3 fatty acids that everybody's talking about. But we have the impression of that, at least.
So
And
they must raise the fries somewhere and throw them into these pens. Is that right?
Yeah, what they do is they have hatcheries. They milk, so to speak, the eggs out of the females and the sperm out of the males. They mix them all together. They have this aerated bin where they keep the eggs until they get eyed, as they call them, and then the eyed eggs are then raised to hatchery level, which is about 2 inches long, which is called a par, and then it grows a little bit more, and that's called a smolt. I might have had that reversed, actually. It's the smolt first and then the par. And then once they get to about 6 inches long, they're moved out into the large ocean-anchored hatchery bins where they feed them. Now, the problem is, what do they feed them?
There's actually some controversy about that whole process. Yes.
Exactly. Lots of it. In fact, they create an ecosystem underneath them from all the waste that they start to produce. And in Norway, where they have these salmon farms in the fjords where the water almost never moves, they've got big problems. If you look at the salmon farms of Maine, there's huge tides and there's an easy clean out for all the wastes, and they don't have a big problem with infectious anemia there. But in Chile, they have lots of fjords there as well. They look just like Norway. In fact, if you go down there, there's all these inlets that go into the Andes Mountains. And so the waste accumulates underneath them, and somehow, this virus infection gets established, and then once it's there, it just doesn't leave.
My understanding is you have to destroy all the salmon.
You do.
You never get
You do.
rid of
The
them.
biggest worry about raising salmon on the East Coast and in Europe also is that they would mix with the wild strains and then dilute the genes. And by the way, there's no evidence for that yet.
There's an article in Bloomberg, and there's also a post on ProMed Mail that this virus also spreads to wild, or as they call them, feral salmon.
Right, exactly.
Feral
But they've always had it. The wild salmon have always had this virus.
Now wait a minute. What's always? Because from what I read that Vince showed up, that Vince sent.
Yeah.
They talk about it showing up in 1984.
Right.
In farms.
Okay.
In farms.
In
farms.
Yeah, but wild salmon, a certain proportion of wild salmon have always carried this virus.
Okay,
Now, in the
that
wild
makes sense.
salmon, does it affect them as severely as it does the farmed salmon, or is this a product of the stress from farming them?
Hard to know.
My feeling is, from what I've read, that the wild aren't affected as much, and also it does infect other kinds of fish like trout, and it doesn't cause any symptoms in
That's
them.
right. That's right.
So maybe an issue with farming.
Right, because clearly we still have salmon swimming around in the seas
Clearly.
wild, and they haven't been wiped out by this.
No.
Yeah.
So all the farms have this now?
yeah,
Basically, that's true. In fact, salmon farming first started in Iceland. And it started as hatcheries alongside of the rivers that led out into the ocean. And all they farmed were the smolts and the par. Then they let the salmon go into the rivers and then out into the ocean. And that's the only farming that they did. And they still had this problem. But, when you let them go and go out into the ocean, you don't actually keep track of how many actually come back. So we don't know what the attrition rate was from the virus versus just predation from other fish or other species eating the salmon once they get out there.
So according to this one source, sea-run brown trout and rainbow trout can be infected in addition to Atlantic salmon,
Right.
but they do not become sick.
That's correct.
So it's just the farmed ones, and that's right. It was first seen in 1984.
Yep.
Now, gentlemen, infectious salmon anemia virus.
Yes.
What family of viruses does this belong to?
I know, but I cheated.
Come on, Rich, tell us.
It's an orthomyxovirus.
Ah,
You just can't get away from
Orthomyxo.
flu these days.
They can't get away from flu. That's right. Here I am trying to find non-flu stories.
That's right.
This is an orthomyxovirus. It's amazing to me. A segmented genome. It's enveloped RNA
Look
virus.
at that.
So that's amazing. It just shows you that particular virus can be in many different hosts. It's not a human influenza virus, of course. It's very, very different, but it's structurally quite similar.
but if they want more funding to study and work on this virus, they should start calling it fish flu.
Fish
Fish flu.
That's right. Well, it's interesting. The symptoms are quite different. You have all kinds of lesions in various organs. And it's not really a respiratory infection as it is.
And you get an
in
anemia,
humans.
presumably.
You get an anemia.
Probably hits the bone marrow somehow.
Let's see. The clinical manifestations in 4 organs: liver, kidney, gut, and gill. So you get manifestations.
So it is respiratory.
Yeah.
What, the gill part?
Sure.
Gil?
Yeah. Okay. So the other interesting aspect is that this is an economic issue. So Walmart stopping to buy from Chile is a big deal.
That's very interesting.
The biggest supplier is Norway. Now, apparently they're getting all the orders now.
That's right.
So Chile's in big trouble. So they're trying to make a vaccine for this. Can you imagine? I don't know how you would administer such a vaccine. I guess you would have newly hatched fish and inoculate them.
Maybe it's a DNA vaccine. Yeah, but they do a lot of that
chuck
stuff.
it in a tank.
Yeah,
Right.
With the fish.
Yeah, you could do that too.
Sure.
So
do a lot of people buy fish at Walmart? It's not really the kinds of products I associate with
The
them.
answer is yes. They buy a lot of produce from these large outlet stores. Costco is another one like that.
Yeah.
I mean, I know at the Walmarts in China, you can buy live turtles and that sort of thing, but I didn't realize that the food market was a big part of the business.
It is, actually.
Well, Sam's Club has a big food section.
That's right.
It's part of the same organization, so
You bet.
You can tell I'm one of those Eastern elitists who doesn't go to Walmart.
I didn't either until I realized I could save money.
Well, I have to tell you, when we got Tamiflu prescriptions in Florida. So, Rich, my son came down with what I thought was flu on the first day of our vacation.
Oh, great.
In Florida. So our pediatrician gave us all Tamiflu. And where did we buy it?
Got to be Walmart.
It's Walmart.
Okay.
Well, you're in Florida, yeah.
We called around and they had it, and they said, "You're in luck. We just got a new shipment because it was all out yesterday."
So did your son have flu?
I don't know, but the next day after one treatment of Tamiflu, he was 100% better. And we were able to go out into the world.
We would call that anecdotal evidence.
So it's anecdotal.
Right. I was gonna say.
But the pediatrician said, "Yeah, you should take Tamiflu and stay in the room for 7 days." And we said, "Sure."
Right.
Problem. So I don't know if he did, and we didn't get him tested, but it's anecdotal. And last week I had some symptoms myself, and I took some Tamiflu, and the next day it was fine. And so it's all anecdotal. I'll never know. I was going to culture some virus from myself, but after a few days of Tamiflu, I felt it wasn't really worthwhile. So,
or as I once heard an infectious disease doc put it, it was an empirical treatment.
Yeah. Well, I didn't have any side effects from the Tamiflu. That's all I can tell you. But anyway, Walmart, is a big deal, and Costco and all these big places. The world. Listen to this. The last thing on this story: the world supply of salmon, 1.3 million metric tons. And this is the farmed salmon, basically. It's going to go down 10% this year because of this Chilean issue, 10%. Oh. So in terms of effects on food, Dick, this is a big deal.
That's true. Well, people will have to eat something else then.
A lot of protein, right?
They'll have to switch their diet.
So it's an interesting virus, and this is obviously a big deal, and we're going to see it more. So I thought we should
time to do some bass fishing.
Chilean bass fishing, that's right. Well, we
bass.
almost extincted that fish, so.
Yeah.
And notice that you can't find orange roughy on the menu anymore either.
No, you can't.
Right.
All right, now our next story also has to do with a very important food, and that's wine produced from grapes. And this is a story about making virus-resistant grapevines.
Aha.
So, Dick, you always wanted to talk about a plant virus.
I did.
So it turns out there is a virus called grapevine fanleaf virus.
But of course.
Which just knocks out grapevines. It's a big problem for wine growers.
Yep.
And this is a RNA virus. It's icosahedral non-enveloped. It has a genome in 2 pieces. There's 2 RNAs in this capsid.
Huh.
And it's spread in a couple of ways among grapevines by grafting. I guess they do that a lot in grapevines, right? They graft pieces.
That's
so
right.
if you're infected, graft.
That's right.
It's spread by mechanical means.
Rich, you're in the heart of the grape country there.
True.
And they make some wonderful Pinot Gris.
Yeah, Rich is not a wine guy.
No?
I'm just kidding.
Well, become one.
By farm equipment and also by nematodes carry the virus from plant to plant. So I just found this this week, and apparently this is a big economic problem.
Sure.
So there's a lot of research to try and fix it. So what they have done is it's a lab in Germany. I found a couple of articles. Stefan Schilberg is the PI. What they did is they found that if you make antibodies to the virus, which plants cannot do, right?
Right.
Plants don't make any. But if you make antibodies in mammalian system and you put them into the plants, it will stop infection. So the antibodies bind the virus and stop infection. So what they're doing is engineering the grapevines to make antibodies.
Right.
To prevent infection.
Right.
And they've done it in a test organism,
Nicotinia.
Nicotinia benthiana.
Or some yeah.
Nicotiana.
Nicotiana, I think it is. Tobacco.
It is.
Tobacco plants, yes.
Correct.
That's the standard plant virologist.
That's right.
That's sort of Viro cells for a plant.
Well,
virologist.
the tobacco
because nobody
mosaic.
protests when you kill them.
That's right.
They actually put the gene encoding the antibody in a bacterium, agrobacteria, which then delivers it to the plant. And they've shown that you can protect them against infection. I think this is just great.
I wonder how the wine tastes, though.
Well, that's an interesting question. Are people going to object to these? Are basically genetically modified wine.
You bet they are.
The wine drinkers are not going to object.
It depends on whether you ask them before or after they open the bottle.
That's true. That's true.
Boy, I'll tell you, if somebody had come to me with that idea, I would have told them they were crazy.
You think?
But what a terrific idea!
Isn't that great? It's it's good virology, so the papers are in pretty good journals, and it's practical. I mean, it's not something that makes you sick. But these last two examples show that economically viruses are very important, and we need people working on this stuff.
Well, I was at the USDA 2 days ago and raised that question with them as to why they don't have a bigger plant virus division, and they threw up their hands and said there's so many of them. There are 1,400 different plant viruses listed on their website. So, and they're all important at some time or another during the history of growing crops. So, take your pick. No matter what you do, you're going to be overwhelmed by the amount of work that you need to do.
Well, there is some, and there's so many other viruses to work on. Also, it's a problem.
Yeah.
But
but one of the first was tobacco mosaic, right?
Sure, it was the first discovered, crystallized, so many different things. Nucleic acid,
Exactly.
It's important stuff, but economically too. So this is great. I was at an ASV years ago in Utah. I was sitting somewhere and I heard guys talking about fish viruses, and I was just fascinated. The same kind of stuff here. It's a big deal in fisheries, and probably not enough work done on it. So all of you budding virologists, agriculturally important viruses, there will always be stuff to do.
That is the point.
Do we have budding virologists?
No, we have budding mycologists.
Mycologists, I know. Yeah.
We do have budding. We also have icosahedral virologists.
That's right.
Okay, we have more stories.
We have a lot of latent virologists.
But before we get to that, we have a word from our friends at Citrix, the makers of GoToMyPC.com. If you're tired of spending late nights in the office or the lab,
boy, I sure am.
It's time for you to get GoToMyPC, because GoToMyPC, which is brought to you by Citrix, lets you access your office computer from home or anywhere you want to be, as long as you have an internet connection. So you could leave work early, have some fun, and finish up the work later when it's convenient for you. It's so easy. You go to gotomypc.com/podcast. A few clicks of your mouse. Within a minute or two, you'll have it installed on your computer, and then you can access that computer from anywhere. And it's completely secure, completely easy and transparent to work on. And you can try GoToMyPC for 30 days for free. Go to gotomypc.com/podcast. That's gotomypc.com/podcast. You'll get a free 30-day trial. We thank them for their support. And now we have a study which was published Monday in Nature. This really kind of raised my hackles a bit. And this is a study of an H1N1, the pandemic H1N1 influenza virus, comparing it with a seasonal H1N1 strain in mice, ferrets, non-human primates, and miniature pigs.
Mini pigs. Oh, yeah.
Pathogen-free mini pigs.
Oh, yeah.
And actually, I got this because a reporter at USA Today sent it to me, a guy that I've been corresponding with. He wanted to know what I thought of it. And there was a quote by the lead author. So there are about 50 authors on this paper, as you can imagine. It's a lot of work. Yeah. And what they find is similar to the results that we discussed last week, where there were two papers published in Science where this virus was examined just in ferrets. And they find it replicates more extensively than seasonal H1N1. It also spreads to the lower tract and grows there well, whereas the seasonal H1N1s don't grow very well. And it causes a little bit more morbidity, so the mice lose weight, the ferrets lose weight. No death, but there's clearly a difference in the pattern of infection in all these animals, with the exception of the miniature pigs. They get infected, but they show no symptoms. So last week, the authors of the Science papers had said, this is pretty consistent with what we see in people. It's mild. It does grow in the lower tract, but It doesn't show as more disease in any way. The author of this study, who happens to be Yoshikawa Oka, he's quoted saying, "There's a misunderstanding about this virus. People think this pathogen may be similar to seasonal flu. This study shows this is not the case. There is clear evidence the virus is different than seasonal flu." And I got sort of annoyed because he seems to be ignoring all the infections in people that are going on and saying, here, my ferrets and mice and pig infections are more relevant. And, I've always, I love animal models for viruses, but I've always said, and I've always been reminded by study sections, they're just models.
That's right.
And they don't tell you what's happening in people. So I don't know what you guys think about this, but my feeling is it's pandemic flu. You get headlines, you get publicity, and everybody wants a piece of the action, and you ignore basic science to a certain extent.
I would agree.
So this gets back to your question, Rich. How virulent is this virus in people? And, so far globally, we've had 400 some odd deaths, millions of infections. It's not terribly virulent as of today. Not talking about going forward, but as of today, my feeling is it's not terribly virulent. Mm-hmm. I think a number of flu virologists will agree with this. So what does it mean if you find it's growing in the lower tract of these lab animals? I don't know if that means anything.
Well, I mean, if you... comparing it to the other strains that are out there, I think you can say in an animal model it's different.
Right.
Okay. But in terms of what that means in humans, you don't know because of the limitations of animal models.
Exactly.
Right. Yeah, I think that's the bottom line. In animals, it's clearly different, but who knows what that means in people. I suspect if you went back and tested a whole slew of seasonal flu strains, you might find some that grew in the lower tract of these animal models, but they never amounted to much in people. Right?
So what would be very interesting to know is how virulent is this strain for birds, because that's actually the definitive host for this virus. So we are animal models for the bird virus.
Yeah, well, nobody's really looking. Everyone's doing this in pigs,
Understood,
and ferrets and mice, but no one really knows. Well,
the flip side is that I think if this virus was as nasty as something like 1918, we'd know it by now.
Oh, yes.
I think so, yeah. You're absolutely right. And people are wondering if it's going to become that nasty. Who knows? But that's not the issue right now. Right now, it isn't terribly nasty.
Nope.
So, this raises another question about what is it for a virus like flu to become something else? Mm-hmm. How many changes does it take? And I think that's a really interesting question. Mm-hmm. And the data that I've seen makes it look like that for to create a new pandemic or to change the tropism of one virus takes multiple changes, and I think that that's an important observation.
Right. Yeah, and we have a really nice opportunity now to track that with this outbreak, right? Because we're collecting samples daily, and we're monitoring continuously. So as soon as it changes in people, if it does in terms of virulence, we'll be able to see what kind of mutations are associated with that. It should be very interesting. Right. But so far...
Meanwhile, H5N1 continues to smolder, and there were some outbreaks in China recently.
Yeah.
So hasn't gone away.
No, I don't think it's going to go away, but what's happening next, who knows? Anyway, this will link to this story. I think it's... I just regret getting people scared by saying things. There were headlines. What was the headline in the paper that I saw? Oh, yeah. Study suggests H1N1 virus more dangerous than suspected. That's just crazy. It's just as dangerous as we can see it right now. Right?
That's right.
400, 500.
We don't have to suspect anything.
Well, the interesting thing came up earlier in this conversation as well, and that is the word pandemic has acquired a really terrifying connotation, and that's not what it is at all.
No.
It doesn't necessarily mean that this virus is going to go out there and kill everybody. It just means that it's a new virus that's spreading worldwide.
That's right.
Absolutely. We should emphasize that all the time.
It's one of those scientific terms that got picked up and misapplied everywhere.
Correct.
Right.
I think that's why WHO is so reluctant to use it. They finally gave in to it, but they took a long time.
Okay, next story. Did a 1918 influenza virus originate in birds or not? Alan, that's yours.
This just came out in science in PNAS recently. It was Robert Webster and his colleagues at St. Jude's in Memphis did this study where it's a computer modeling study. They took sequences of pretty much all the flu isolate sequences that they could get a hold of. Going back into the 1930s, and then also the 1918 strain, which we have some sequence information from, but not obviously a large number of samples. They fed all this into the computer to calculate the evolutionary relationships between these different strains. And what they were trying to figure out was not only, where did these viruses come from, but where should we expect new ones to come from? And one of the long-standing contentions or recent contentions has been that 1918 virus represented a jump directly from birds into humans rather than going through an intermediate like pigs, and that's what made it more virulent. This study seems to undercut that. The evolutionary tree that they came up with suggests that, in fact, the 1918 strain would have been circulating for some time, possibly in pigs before then, undergoing additional reassortments. And they actually project that they think it made the jump to humans long before the pandemic began and was exchanging through humans and pigs for some time before it actually got to the point of causing the 1918 pandemic. Now, this obviously, the usual cautions apply. We're dealing with the very, very small sample sizes as you go further back in time, and we really don't have a lot of information about 1918. But I think it's interesting. It's an interesting highlight that kind of shows, well, we don't know exactly where the 1918 flu came from. It may have come through something of a conventional pathway of what we think of as flu jumping from birds into swine and then into humans.
Yeah, it's too bad we don't have more samples from then, but that's really going to limit it. Taubenberger says exactly what you said. We have so few samples that we'll never know what really happened before.
But I loved in the Science did a little news piece on this, and they interviewed the lead author, Webster, and they conclude, in addition, surveillance in pigs should be expanded, says Webster, although he doubts that anyone will make the money available to do so.
I don't think that's right. I think that surveillance in pigs will increase. I mean, especially because of this year, right?
But there's going to be a lot of resistance to that. I mean, look at what happened with mad cow disease.
Yeah, the pig farmers, basically, right?
Yeah, the pig farmers are not going to be keen to go testing for a virus that they're going to find.
Right.
So if you show up on a pig farm and say, I'd like to test all your pigs for flu, you're not going to be welcomed with open arms?
Probably not.
Yeah, it's too bad.
The notion that at least some of those sequences were in humans or in and out of humans or whatever before 1918 is, I think, really interesting because it suggests that some of the evolution may have gone on in humans, and that's relevant to what we're watching today with the new H1N1 virus.
Yeah.
Right.
It's possible that someday we'll find frozen stuff, right? We just found frozen mammoths that they use to sequence genomes.
Although there's less and less frozen stuff every year.
This is all
true. Yeah, we have to hurry up, right? But there is a lot of frozen material still, so maybe we need to go up there and do some permafrosting. Anyway, that's cool. And the next story is also yours. Holy water ban to halt swine flu.
Yes.
That's a good one.
Yes, the Bishop of Chelmsford in the UK has sent out advice to the parishes to discourage the use of holy water. They in setting out bowls of holy water or stoups of holy water, I guess they're called, for people to make the sign of the cross. I'm not Catholic, so I don't know exactly all the terminology that goes with this. But
you've got it right so far.
It's good so far.
I used to be Catholic.
So he's advising that maybe you shouldn't set out these bowls because it might speed the spread of flu in your congregation, and he's suggesting that maybe people should wear face masks if there's flu in the congregation as well.
Yeah. I'd be more worried about West Nile virus mosquitoes breeding in
Yes.
Yeah, that's right.
Now, if I'm not mistaken, at least they shouldn't worry about the communion stuff because through transubstantiation, that just becomes something else entirely anyway.
That's right.
You're going to get a lot of mail, Alan. And none of it will be good, I can guarantee you.
Well, all they have to do is put a little bleach in the water and it would be okay. I found a couple of papers that showed that just need a little bit of bleach, not much, and it will inactivate the virus.
A box of rubber gloves.
Holy Clorox, Batman.
Holy Clorox. It's a funny story. It's true, though. The water is a good way. So when you go into a Catholic church, there's holy water there, and many people that you dip your hand and you make the sign of the cross. It's a great way to spread, absolutely. And the virus will survive in water depending on the strain. Especially the avian strains survive a long time in water. So it does make sense. And I like what he said here. He said we should also not drink wine from the chalice. Especially if you have flu-like symptoms.
Only the bishop gets to drink wine.
So that makes perfect sense. It's a good idea. Anyway, in looking at this article, I found a bunch of interesting studies on, how flu lasts on your hands and washing and all. It lasts on currency for a couple of days, actually. It's been shown.
Gives new meaning to becoming a viral host.
Host, you mean host, right?
Yeah, exactly, right.
Okay.
Alan, you're definitely going to get mail.
All right, the next paper is a little throwback to the last time Rich was with us. It's called Serologic Evidence of Frequent Human Infection with WU and KI polyomaviruses. And you may remember, Rich, last time you were with us, we talked about a PLOS paper from Bob Garcia's lab where they had done serosurveys for the prevalence of antibodies to these polyomaviruses in humans. And they found pretty widespread seroprevalence and increasing as you get older to BK, JC, and a bunch of new polyomas. And these are viruses that, for the most part, don't seem to be causing disease, but they're with us. So we'd like to know to what extent they're with us. And this paper really just confirms and extends the findings. These samples are from St. Louis, 419 patients. Similar findings, very high seropositivity for both of these viruses.
Now, if you're seropositive for both WU and KI, do you become a Wookiee?
Wow.
Alan?
That's a good one.
Alan.
Wookiee.
You can turn
These
off
are
your
deep
microphone.
questions. These are really deep questions.
That's a good one. You're a Wookiee.
So
Well,
let me ask-
I looked at that paper, and one of the things I liked about it was that they did the age distribution of the antibodies, and there was a peak at less than 6 months, and then a dip between 6 months and a year or two or something like that, and then it rose up to some sort of 75 or 80%. And so that says that there's maternal antibody to this thing.
Yep.
And then you catch it, the real infection, and when you're about a year or two old, something like that.
So for us novices, could you just enumerate for us the most dangerous polyoma type viruses?
In a normal situation, none of them are really dangerous. As Vince said, we catch these things, and
None.
they establish a persistent infection in the kidneys and just hang out there for the rest of our lives.
Now, that's not what you put in your grant proposals, though.
No, what you put in your grant proposals is that if you're immunosuppressed for any reason, and actually it's interesting, one of the things that interests me that I don't understand is that it looks like different types of immunosuppression induce different viruses to do nasty things. So JC virus infects the brain in AIDS patients, causes a disease called progressive multifocal leukoencephalopathy, PML. That's a demyelinating disease, I think, and is a very common opportunistic infection in AIDS patients. Ultimately, it's a significant cause of mortality in AIDS patients. On the other hand, bone marrow transplants, BK virus often comes out and establishes a nasty infection in the kidneys and the bladder, and can cause hemorrhagic cystitis and can be a real big problem. So I don't know why there should be a difference of these two viruses homing to different places under different conditions of immunosuppression. But those are the two nasty ones. Hemorrhagic cystitis with BK and PML with JC.
Yeah, the WU and the KI, is, according to this paper, have not been associated with disease yet.
Nope, and I'll bet you there's more out there.
Sure.
I'm sure you're right.
No, the Merkel is associated with Merkel cell tumors.
Yeah, and I didn't understand what that disease was. I didn't look into that. What is that?
I don't know offhand.
Okay, we'll go find out.
Are there any animal diseases that polyoma causes?
Well, there's polyomaviruses in a whole bunch of different animals. The reason it's called polyoma is the guy who discovered it, and the name escapes me at this point, was actually looking in mice to see if he could find a virus that caused mammary tumors.
Right.
So he was making extracts and passaging them in mice, and he found some sort of filterable agent that caused all sorts of tumors.
Right.
Polyoma in newborn mice, and that turns out to be a mouse polyomavirus that ordinarily infects mice, just like humans, doesn't cause any problems. But if you infect newborns that don't have with enough virus, that don't have an established immune system, or a heterologous host, you can see a problem. Subsequently, there was a paper where I'm thinking the author is Rowe, and I don't know if that's Wallace Rowe or not, but at any rate, went out into Bronx or somewhere in a bunch of abandoned projects and assayed mouse colonies for antibodies to polyomavirus, and also did whatever sort of clinical stuff that he could on these, and established that this virus was a natural pathogen in mice, didn't cause any disease. Some colonies had it, other colonies didn't.
Well,
Fascinating.
Okay, Rich, you're using the word pathogen, but it doesn't cause any disease, so I would, caution you to say it's an infection.
Okay.
That could lead to disease. I'm sorry, I just...
No, that's fine. I stand corrected.
Because I'm not a virologist, but at least I can give you my 2 cents worth on that level.
So, Rich, a Merkel cell carcinoma is a rare type of skin cancer, also known as neuroendocrine carcinoma of the skin, mostly develops in older people. And Chang and Moore, who discovered Kaposi's sarcoma virus a few years ago, published a paper suggesting that this new polyomavirus is associated with these carcinomas. The association is quite good.
Interesting.
And so that's what that is.
Sun exposure and immunosuppression seem to be the main risk factors, and but it is rare.
Yep, quite rare. So that's pretty neat, and we'll see more of this, I'm sure, as we scan more. But the thing, I want to close just with this on this topic. We know to look for these viruses, but how many are out there that we don't know to look for yet? That's the thing.
The unknown unknowns.
As Rich said, there's more. There's more out there.
Sure.
Just have to find them.
Of course.
Our next story I thought was also interesting: reemerging rabies and lack of systemic surveillance in the People's Republic of China. Now, 2 things interesting here. First of all, the record of rabies in Chinese history dates back to 556 BC. In Master Zhuo's tradition of the Spring and Autumn Annals, he writes, in the 11th month, people in the capital of Song were chasing a rabid dog. And so I didn't know that. It was so far back in history. And the second is that there is currently a rabies epidemic in China. It's reemerging. And this paper highlights the issues. There's a growing dog population in China, and there's not a good immunization program for pets. And so therefore, they try and kill the dogs, but that never works, right? You can't do that. And they say in this article, basically, you should be... We don't have our own vaccine for pets. We have a human vaccine, but you have to have a pet vaccine because that's how you stop the spread of the virus. And there isn't a good diagnostic program either. So I thought it was interesting here in China, economically very powerful emerging nation emerged, and they have a rabies problem.
Yep.
It shouldn't be.
The most dogs I ever saw per square mile was in Bhutan. There must have been 20 dogs per person.
Wow.
And they were all running wild, and they all barked at night and kept everybody up. And they were mangy and very underfed, and the weakest dogs were not around the next morning. And you know that expression, it's a dog-eat-dog world. Well, if you want to go see that in action, go to Bhutan.
More than 50,000 people die each year from rabies globally.
It's a big disease.
In China, they say here 100,000 people have died of rabies since 1950.
It's a horrible death.
A lot of people. They shouldn't be dying because the vaccine is very good.
It's very easy, and it's an oral bait that you could feed to these dogs, and immunize them orally.
So they also say that they don't have a good laboratory system for diagnosing infections in China, and they need to have that as well. There's a lot of inconsistency, and they say there are bootleg vaccines which don't work. So you give it to your pet, and it doesn't do any good. So they say in the outlying, not in the cities, but in the countrysides where the population is large, they're getting more and more dogs, and this is a problem.
Right.
So I like this article because it tells you pretty straightforward what's going on and what has to be done there. Yeah. So here, in contrast to human rabies vaccine development, animal rabies vaccine development in China has not progressed. In the U.S. alone, there are 11 different rabies vaccines licensed for dogs, 12 for cats, 1 for ferrets, 3 for horses, 4 for cattle, and 5 for sheep. In China, there is 1 vaccine licensed for pets. So it's a big difference. And that's the way you control it. Not only in dogs, of course, but eventually in wild animals. So they're going to have to get to that point, right?
They will.
They do the baiting with the vaccine.
Right. Yeah, in populated areas, dogs are the major reservoir of rabies, no matter where you look. Whereas in the wild, it's all sorts of other animals.
In New York State, it's raccoons. That's for sure.
They say here that you need greater than 70% coverage of pets to sufficiently control rabies.
Right.
And so they're not even near to that. So,
and with the issue of stray dogs that's been discussed, that's a mammoth task.
Absolutely.
Huge. Yeah.
It's interesting. I can just interject something here because I'm aware of this program. The United States military is involved in a rabies control program all along the U.S.-Mexico border because they patrol it regularly for other things. They drop oral baits out of the helicopters, and it's to prevent coyotes that migrate up and down between the United States and Mexico from bringing rabies up into the U.S. from Mexico, and it works. It apparently works very well. So you can monitor very large borders if you have the inclination.
And money.
And money. And China has both of those things, by the way. They've got tons of money and tons of inclination. I'm surprised they don't just hire some of these vaccine developers in the United States to come over and help solve their problems.
Well, and they have personnel too. They wouldn't necessarily have to drop these baits from helicopters. You can have 10,000 people go out in the street and distribute these things.
This is all true.
And I don't know. I didn't realize that there were so many vaccines available. The only one I'm familiar with, I discovered when I had my cat vaccinated, and that's a canary pox that expresses the rabies glycoprotein.
Right.
And I think they use that in these baiting situations in the wild as well.
They do work, though, because
they do.
Throughout Europe, it's worked beautifully.
Yeah, sure. No, the pet immunization also works very well. Mm-hmm. I didn't realize that you needed a different one for each species, though. It's interesting, right?
Maybe it's because certain pets don't eat certain foods, and you have to put them in different things.
Your pets are injected. Those are all injected vaccines.
Are they?
Aren't they? Isn't that right, Rich? Your cat probably got injected.
Yeah, it's injected. But actually, the baiting situation, that's interesting. I read an article one time about developing the bait for these animals to eat. That's a whole science in itself.
Yeah, right.
These guys in the laboratory trying to get something that the raccoons will actually eat.
Exactly. You have to make it crayfish-like.
Yeah, I think one of the innovations there was to have little plastic capsules that the vaccine is actually inside the bait, so that the raccoon or the dog teeth penetrate the capsule and release the vaccine as it's eating it. So the vaccine prep doesn't contaminate the flavor because they're very sensitive to smell.
That's true.
Okay, a couple more stories. All very interesting. I don't want to cut any of them out, but we're starting to run close to the limit here.
Yes, we are.
The next one is increased host species diversity and decreased prevalence of Nombre virus. This is
they really should give it a name.
Dick, this is up your alley.
Yeah, well,
this is very cool.
One of the flavivirus.
So this is in the Emerging Infectious Diseases Journal.
Oh, yes.
Basically, the hypothesis is for Lyme disease, for example, the more species the organism infects, you get a dilution of infectivity, and there's less disease overall. So they wanted to address this hypothesis for Nombre, which is a hantavirus, causes in people hantavirus pulmonary syndrome, right? It's a recently emerged virus. So they went out into the field and looked in several environments. They measured the species diversity of the virus. In other words, what kind of animals is the virus present in? Trap different kinds of animals and look for virus. And what they found basically is that when it's present in more animals, the diversity, the amount of virus detectable goes down. So the more species in a certain ecological area dilutes it.
Yeah.
You know about this, Dick?
I know something about that. I mean, I misspoke before by calling this a flavi. It's not a flavivirus, obviously, but I thought you were going to refer that to the West Nile virus, which has one of the broadest host-specific ranges that you can possibly imagine. It seems as though the wider the spread of a virus infection throughout the animal kingdom, the less virulent it is. It does seem that way.
I don't know why it would be, because you would imagine that it still replicates well in the normal host. So in the case of the Nombre, the Paramyscus maniculatus, if that species is present, why wouldn't it?
Well, by serially transferring viruses, that's the way you attenuate them anyway, isn't it? So maybe serially transfer them from one species to another, maybe that diminishes their ability to cause disease, but maybe it increases their ability to infect, which would favor the virus in this case.
What they suggest is that when you have an increase in species diversity, you get more predators of the actual hosts of the virus, and that could eliminate them as well.
Oh, I see what you mean.
That could factor
when
in.
They get sick and slow down, then they get caught.
Yeah. So basically, it's very interesting. And one of their main arguments for doing this is that they say speciation is decreasing in most parts of the world.
It is.
Right? We're getting fewer and fewer animals in Africa, all these places where viruses are emerging.
Decreasing the biodiversity index.
Yes, so that is bad because the virus prevalence is going to increase. Right?
Aha.
Right.
So that's the significance of this.
This is one prediction that you could make on the basis of this. That's true.
Right. So
one of the things I love about the whole hantavirus story is that when that broke and they discovered what the host was, a whole bunch of research that went on for decades before looking at the ecology of these mice and the different species of mice all of a sudden became relevant.
That's right.
Okay?
They could explain
Not that it wasn't relevant before, but relevant in a different context.
Oh, you're quite right.
Okay? It's just fascinating.
You can't imagine how freaked out I was at one camping trip when a white-footed mouse jumped out of my... Oh my God,
Yeah, that's a good point, though. I mean, that just goes to show you have to do research, and you never know why.
That's right.
It's going to be. You can't do everything, though. Obviously, right? You have to make decisions. But in that case, it was fortuitous that it had been done.
It pays to know the old literature.
I mean, this kind of study is quite extensive. They have to go out and trap various animals.
Absolutely.
Look for virus by PCR.
It's underappreciated work, I would say.
It is underappreciated. And even this work, a layperson might say, why is this important? And well, we're trying to understand why we have so many new emerging viruses. And there are lots of reasons we give, encroachment on wild animal populations and so forth. But this is another one where you decrease the number of animals. You shouldn't do that. I don't know how you solve it, though.
Haha.
Right?
That's right.
Decrease the number of humans.
Eventually, that's exactly what's going to happen.
There are some people who are working on that. But I think it's an all or none thing, Rich, unfortunately.
Right.
Yeah.
And so it's not a good thing. Well, we have a couple of great stories left, but I think I'm going to hold them for next time and move on to some email and wrap this up, because I do understand that people don't have forever to listen to TWiV. So we should keep it. Yeah, that's right. So just a couple of emails. Here's actually one I found a note on Twitter. And Adam wrote, I can't sleep, so I'm listening to This Week in Virology.
Yeah, I didn't get that. Is that trying to make him go? He's trying to do that to make him go to sleep.
Yeah, I had the same thought. Did it put you to sleep in the end?
Or I had nothing else better to do, so I had to listen to
I don't know. When I was a graduate student, we always used to read our microbiology textbooks to fall asleep.
Yeah.
That always did it. So he says, good stuff. That's nice. Well, I'm glad you listened to it.
Yeah.
Now, Peter wrote, Peter is a equine veterinary surgeon from Australia, and he writes, Hi, Vincent. Love your show. Keep up the good work. By way of quick introduction, I was the veterinarian involved with the outbreak and deaths of 14 racehorses back in 1994 here in Brisbane, Australia, which was caused by a then unknown paramyxovirus now called Hendra virus. So this is the guy who did it. Isn't that great?
Wow.
He discovered TWiV.
Maybe we should interview him sometime.
I said we want to get him on. I offered to get him on. Absolutely.
Good eye.
For sure.
Good eye.
The time difference is going to make it tricky. but we'll do that. I
This
would
is
love to do that.
pre-recorded.
I have a question regarding serosurveillance for evidence of henipavirus infection of fruit bats in the Americas. In light of the seroprevalence of henipavirus antibody in fruit bat populations here in Australia and also in Southeast Asia, Bangladesh, and West Africa, do you know if serosurveillance of fruit bat populations for evidence of henipavirus antibody has been performed in South America or in the Caribbean, and if so, the results? I looked. I didn't find anything. You know of anything, Dick? Nothing. It would be interesting to do because that's one of the few places where they haven't looked.
This is true.
Apparently,
That,
but I didn't
that would
find
be a
anything.
good study to do.
And then he asks, I believe there are fruit bats from Jamaica and South America and insectivorous bats housed at the San Diego Zoo. Would this provide a convenient captive population for CDC to initiate a serosurvey?
Why not?
These are bats that are captured and brought into the zoo?
Yeah.
They're not bred there.
No. There's one over here in the Bronx also
It should be looked at, I would think.
called The World of Darkness. You can actually go in and watch bats
Yes.
feeding. It's quite interesting, actually.
Yeah. No, I've been to some zoos that have great bat
I knew when I was at Rockefeller, I knew one of the great bat
collections.
biologists. His name was Dr. Griffith,
Uh-huh.
and he was a big man on echolocation.
I think it's a good idea, but I think the surveys in South America should be done. Looking
Bats have
forward.
got everything. They're a reservoir
They
for
do.
all kinds of
And
stuff.
remember what we said very early on in TWiV, there 20% of the mammalian species are bats.
Is that right?
That is correct.
Wow.
They are to mammals what beetles are to insects.
Here, here.
But they're having some issues with white nose
They are.
disease, right?
They are.
Yes.
Now,
That's
Dick,
true.
what's going to happen if we have a 90% decrease in the bat population?
In this country, I think you're going to see a burgeoning mosquito population, and then more disease transmission. That's what I think would happen. I think they're great at controlling diseases by eating insects. I'll give you the example of that. In China, I know there's no time for this, but I'll just take it anyway. In China, in the main squares of Beijing, they decided that the sparrow populations were causing too much damage to their statuary by defecating on them. So they decided to mist-net them out of the environment, and they did. And the next year, they had one of the biggest outbreaks of encephalitis they've ever had. And they decided, perhaps the mosquito control program initiated by sparrows is what we should try to maintain. That service that the sparrows perform is called an ecological service, and we are busy disconnecting those services left and right. So bats perform a great ecological service by eating all these noxious insects. So, we don't realize how good we have it until we knock something out.
So he ends up by writing, looking forward to your reply and would like to see you do a program on these newly emerged zoonotic diseases, Hendra and Nipah. Let me know if you need any input.
Wow.
Yes, we do. We're going
Sure,
to have you on the
absolutely.
show. We'd love
You
to
betcha.
have you on the show. That would be great. Liz wrote, first, thank you for podcasting TWiV. I'm a science enthusiast with an amateur level of knowledge. TWiV has been a great learning tool. What do you make of these findings? And she actually links to the Nature paper that we just talked about.
Huh.
She says, I'm alarmed by the headlines hinting and the media insistence that this is a 1918 redux in the making. Regarding the lung lesions, increased severity of disease, I'm guessing that the diversity of animal test subjects indicates that there would be similar results in humans? No. So you
Right.
probably can just go back and listen to what we've said, Liz, but it doesn't mean anything about people.
Right.
It's an animal model. So don't be alarmed. So that's what it bothers me about.
Yeah.
This one thing I didn't point out, which I wanted to ask you guys. So this Nature paper, which was rushed to publication, basically takes two viruses and puts them in three different animals. Do you think that could ever be published with any other virus at another time?
No.
Not in nature.
Right. Not in Nature. That is not a Nature paper. And the only reason it is because it's the pandemic strain, right? I have tried to do 10 times more than that and get it, and Nature doesn't even look at it.
That's because you don't have an English surname.
Yeah, okay. Well, I just wanted to. I don't mean to say that they shouldn't, but it's not a Nature paper.
Nope.
Okay, Jim. All right, here's a cool one from Jim. Jim wrote us a long email a long time ago because he went through every episode and wrote down questions. And I got one question for today, and I thought Rich would love this one. Vince and Dick, if one virus penetrates one cell and it generates from 100,000 to 1 million offspring, why does it take more than one to overwhelm a system?
So I didn't quite understand that question, actually.
I think he's asking how many viral units are infectious to begin with.
Well, if one virus is enough to infect the cell, and you make a lot. So then
The assumption is wrong. One virus is not enough to infect a cell. You need about 10, and one of them will probably
that's
succeed.
part of it, yeah. Usually
I think the infectious dose for any infectious agent is not 1.
Right.
I don't know of any that's 1. Do you?
No, in cells sometimes 1 is enough.
Maybe rabies.
For some viruses.
But
in animals, it's probably much more than 1.
Same in parasites.
And then you have the complication of an immune response that kicks in very quickly, an innate response that's right. Does a lot of dampening.
Sure.
What do you think, Rich?
And as was pointed out, viruses are also they're slobs. Okay,
they are.
They make a lot of dead particles, leave a lot of trash behind, and that kind of stuff. But as long as you make enough infectivity to move the thing forward, you're okay.
Right. They do make a lot of trash, but they do well.
We're talking trash
That's
here.
part of their beauty that they make so much. That's why they evolve.
I guess.
Then Jim sent a link to a story on developing a fast, cheap PCR test for crime scene analysis. And he asked, could this lead to home tests for flu that users could then report to some collector? So I don't think it's still a PCR. I looked at this test. It's still a PCR. I think that's too much for home. But we had actually a story about a really cool diagnostic test. We'll do that next week that involves things that you could do at home. And so I think there are ways to do that, Jim. There are home ways that you could test, but not by PCR. And of course, Jim, you always have a problem when someone does a test at home, you don't know how reliable it is.
That's
right?
correct.
The specimen collection may not be appropriate. Running
Could
the
be
test.
a false positive.
I'm actually more optimistic about this.
Yeah.
I think, particularly with these lab-on-a-chip systems, the microfluidics, I don't know if you guys have followed this trend, but there are several researchers and companies now who are using surplus equipment from the silicon chip industry to etch channels onto chips, and you can direct things through the chip by capillary flow. And of course, since it's a silicon chip, you can also put in wiring, and you can actually build a little thermal cycler on a chip. So that you could
nanotechnology,
right? And you can mass produce these things, so you could potentially have a test that takes some kind of sample, takes it in and does a PCR reaction, or does some other assay, does an antibody-based assay, and then produces some kind of a readout. And as far as home testing goes, I mean, there are home HIV tests, there are home pregnancy tests, and the key is just designing a product that is, well, nothing is ever foolproof, but sufficiently idiot-resistant that even if it's misused, you'd have some kind of internal controls on it that would indicate, okay, the test worked and the result is this, or the test didn't work, therefore the result is meaningless.
Yeah. No,
so I think it could be done. It's a question of whether somebody's going to do it right. I
think you're right. I'm thinking about current technology, which is even in a doctor's office isn't 100% reliable. But
sure, it is not just around the corner.
Yeah, I agree though that the future, you're going to get up in the morning and stand in front of a sensor, and it'll tell you everything. I really think
it'll tell you if you feel okay.
And that's right. Report all that information to
I want one that says you look like $1 million.
Green and wrinkles. Yes.
Not in my lifetime.
Lose 10 pounds.
I don't know if you guys remember Star Trek. The physician had a scanner. He would scan someone.
It's a tricorder.
Tricorder. I think that's going to happen someday, and it will tell you all about your virus and bacterial and fungal infections.
You bet.
Well. But we had a story which we'll do next week, which is a really cool detector using more sophisticated approaches. And this is actually a melding of virology and engineering, which is what you need to develop this stuff. So we'll talk about it next week. We'll do one more and have some picks, and that'll do it. And this is from Lisa. I'm a registered nurse in San Diego. Through the Science Podcaster Network, I have discovered and listened to all of the TWiV podcasts. Wow. Aside from that, I have read many of the blog postings and examined many of the links that you and your co-hosts have recommended. As if I had any more free time aside from my 2 jobs and all of the listening and reading that I do, I bought your double-volume textbook.
Good Lord!
And I'm slowly working my way through it. Good for you. My learning curve is huge, as is the textbook. I often find myself needing to go back to other sources to refresh my memory on some of the basics to fully appreciate what you have written. It is a marvelous thing, that book of yours. The field has advanced so much since I was last in a biology class. I'm 52 now. When I hear some of your readers' questions, I now think that they too would benefit from the purchase as well. just so that they know it isn't completely dry, you do throw in some tongue-in-cheek comments, which I really appreciate. Also, TWiV and podcasts like it have compelled me to go back to school and finish my BSN.
My goodness.
And I thought that just buying the book was expensive. This podcast of yours is going to cost me a fortune. But knowledge and healthful living are the best investments. This morning, I was watching a program on our local university channel about using Google as a research engine. The speaker mentioned looking books up on Google Books. I put in one of your weekly picks, Fever by John Fuller. As of yet, there are no reviews for the Google site. I just thought that I'd let you know since your show had recommended it. Perhaps if TWiV got on this particular Google site to add to reviews of your TWiV recommended books, it would promote your efforts even further.
A nice idea.
I will be buying that book, by the way. It will be my reward when I finish Principles of Virology. I look forward to listening to all of your upcoming programs. Yours and the other programs on the Science Podcasters Network have truly revitalized my career and therefore my life. At some point in the future, I hope to work in the area of viral surveillance for the zoonotic viruses that contribute to the pathogenesis of humanity. I already have a great veterinary contact at Wild Animal Park to help guide me. From your lips to our ears, Lisa. Wow, cool. That's great.
What a nice upbeat letter. That's great.
That's outstanding.
I like hearing how it affects people and makes them more interested in science and so forth. So that's what we want to do.
Rewarding. That's good.
I mean, that's what we're trying to do here is get people interested. So I'm glad to hear that.
Yeah.
And that should do it for email. We have plenty more, but we'll do that next time. And I want to just end up with our science picks of the week. And probably Dick doesn't. You have a science pick?
Of course I do. I think we've neglected to actually give this as a science pick, but this is the year to do it. Origin of Species. You have to read it. You got to read it. It is a must for everybody to read that book. It's a dry book. It's boring, but it's very exciting at the same time.
Good.
It's clumsy because it's written in the style of 18th century England. But it's a marvelous, marvelous, marvelous book. And to say that you've read that book is an accomplishment for every human being, I think. To understand our origins is the whole idea here, and this gives you the best pick of it.
All right. The Origin of Species by Chuck Darwin.
Yep. Charlie.
Of the year.
Charlie knew what he was talking about.
Yes, Rich, you have anything for us?
I got a list, but I'll start with the top of my list and see if this is a new pick. How about The Making of the Atomic Bomb by Richard Rowe?
Cool.
Good.
That's probably the best science book I've ever read. It's absolutely fantastic. Wow. It's huge. It's thick. It's about 1,000 pages, but it's riveting. And it starts from the beginning and goes starts from the beginning and goes through all the way to the development of the bomb. And it's interesting because there's sort of two parallel tracks in the book. One is technically describing what's going on, and the other are sort of mini biographies of all the characters involved. Okay?
Wow.
And
sounds good.
Absolutely fascinating, and ends with this guy's a journalist, right? He's not a scientist, okay? But it ends with a really nice sort of perspective on the relationship between science and society that I won't give away from you, but it's absolutely terrific.
Cool. That's great. I'm going to get that one.
I mean, here we are sitting at Columbia University, and you just mentioned this. I know that you know the relationship between the Manhattan Project and Columbia University, but we played a huge role in the calculations aspect of that project.
All right. We'll save the others for your future TWiVs because you'll be back. Alan.
Yep, my pick is also a book. It's one, again, I just recently moved and I was reorganizing the bookshelf and looking at all these books that I was either going to have to get rid of or move. And this one I chose to hang on to. It's called For Love of Insects.
Oh, I know that book.
By Thomas Eisner.
That's a fabulous book.
It really is a fascinating
you're right.
Slice of a life in science.
Here.
Eisner is an entomologist at Cornell, and he's been in that field for decades. I, like Dick, I have a side interest in entomology. In my case, it runs in the family, but
sometimes it flies, sometimes it walks, sometimes it crawls.
Exactly. No, there is a species, Culicoides dovey, that my grandfather named.
No kidding.
Disadvantage of having an entomologist in the family is that you get a noxious biting midge named after you.
It's a vector.
It is. But For Love of Insects, Thomas Eisner, and one of the really neat things about the book is he goes through a set of, it's kind of highlights of his work, but he follows each of the discoveries through from his initial observation, which in entomology, a lot of the time, one of the nice things is that there's not a lot of technology between the scientist and the observation initially. It's just him walking in the woods or in the jungle, and he sees some insect doing some interesting thing or some other arthropod doing some interesting thing and wonders what's going on.
Right.
And so then he takes this back to the laboratory and explores it and finds out, that millipedes produce cyanide in order to repel ants, and he finds how bombardier beetles work, and just all these fascinating adaptations, and it's a really, really fun book.
Cool. He's great also as a teacher. I've seen him on the Discovery Channel a lot, so he's a fabulous educator as well. And he wrote another book called Chemical Ecology, which is a fantastic short take on all of these wonderful compounds that invertebrates make and how they use them in nature.
All right, that's great. Yeah, I know Tom Eisner. He was at Cornell. He still is, I believe.
Yes.
And that's where I spent a few years, so I knew of him. Well, my pick is actually a suggestion from one of our listeners, Zach, and I haven't used it, but I'll tell you why. It's a telescope. And it's a complete usable telescope for $15 plus shipping. Needs a tripod. Perfect for getting kids interested in science. This is according to Zach. And I put this because it's the summer. It's a great time to be looking in the heavens. I happen to have a telescope, and I love looking up there. Last summer, Jupiter was visible, and I could see some of the moons. And you don't see a lot of detail, but just seeing a planet and moons up there is really inspiring. So
no question.
This is the time to be getting a telescope, and if you got $15, check it out. And that's a suggestion of Zach. Okay, so those are our picks, and that will do it for another TWiV. Rich, I really want to thank you for coming on TWiV during your vacation. Really appreciate it.
Anytime. I love it. Anytime. It's really great.
We'll have you back because I think you get the TWiV shtick. And you like explaining virology, so we'll have you back in the fall once you're back.
Thanks. I look forward to it.
And maybe next year you'll join us at ASV.
Oh, I'd love to do that. That'd be good.
That'd be a lot of fun. And Alan, thank you.
Always a pleasure.
Keep working on those walls of that new house.
Watch your thumb, Alan.
Yeah, watch your thumb.
Yes.
And watch your Catholic comments.
You can follow Alan on Twitter, Alan Dove, or you can read his blog at dovedocs.com.
Yes.
Dick, thank you.
Vince, pleasure always.
Always. Someone asked in an email, which we didn't get to, they said, I have Vincent's and Alan's Twitter, but what's Dick's? But
doesn't have a Twitter.
Dick doesn't have a Twitter.
I do have a website that they could go to called
Which one?
Trichinella.org.
Okay.
Trichinella.org. That's what I used to do my research on.
All right, we'll link that this week.
Good.
Thanks to you. And that will do it. Don't forget we have a website, twiv.tv, where you can find all our show notes. You can send us questions. We love them, as you can see. You can send us an email, twiv@twiv.tv.
And corrections.
Corrections, of course.
Corrections.
Let us know what we missed.
Yeah.
twiv@twiv.tv. You could send us an audio file, an MP3 file by email, or you can phone up TWiV Podcast on Skype. You'll get the voicemail, and you have 10 minutes to leave a message, which we can then play on the show. And nobody's taken that up yet. I guess people don't want us to hear their voices, but we'd be happy to hear what you sound like. Gives a little personality to the emails, although some of them are pretty interesting.
They are.
They are. Thanks, everyone, for listening. We'll be back next week. This has been This Week in Virology, the show all about viruses. Another TWiV is viral.